A new method of 3D printing has been developed by scientists that holds great potential for the treatment of brain injuries. Through the utilization of 3D printing technology, a team of researchers has successfully engineered a simplified cerebral cortex tissue by using human stem cells. Once implanted into slices of mouse brain, the structures were able to seamlessly integrate with the host tissue. In the future, the innovative methodology created by experts at the University of Oxford may offer custom-made solutions for individuals who have sustained brain damage. These professionals have successfully illustrated that it is possible to 3D print neural cells to resemble the structure of the cerebral cortex, which has never been done before. This noteworthy achievement has been disclosed in the journal Nature Communications and is a significant step forward in brain injury treatment. When the brain experiences injury, whether from trauma, stroke, or surgery for brain tumors, the damage often occurs in the cerebral cortex, which is the outermost layer of the brain. This damage results in significant challenges with communication, movement, and cognitive function. For instance, every year, approximately 70 million individuals worldwide endure traumatic brain injuries, TBI, with 5 million of these cases being severe or leading to death. Sadly, there are no viable treatments for severe brain injuries at present, and this lack of effective treatment leads to grave consequences for an individual's quality of life. In the future, tissue regenerative treatments may provide a hopeful solution for brain injuries, particularly those that employ implants created from a patient's own stem cells. However, up until now, there has been no technique that guarantees that the implanted stem cells replicate the intricate framework of the brain. The University of Oxford conducted a recent study where their researchers fabricated a brain tissue with 2 layers by using 3D printing technology with human neural stem cells. Upon implantation into brain slices of mice, the cells displayed a notable level of integration in both form and function with the host tissue. According to the primary author, Dr. Yongcheng Jin, who is a member of the Department of Chemistry at the University of Oxford, this breakthrough signifies a major accomplishment in the quest to create materials that imitate natural brain tissue structure and capabilities. This achievement will enable researchers to investigate the inner workings of the human cortex in ways not previously possible. Furthermore, in the future, it may provide hope for those who suffer from brain injuries. Human-induced pluripotent stem cells, iPSCs, were used to construct the cortical structure, and these cells are known to have the capability to produce the various cell types that are present within most human tissues. The use of iPSCs in repairing tissue has a significant benefit in that it can be harvested from the patients themselves, which means that there is no risk of an immune response being triggered. Using a combination of chemicals and growth factors, the iPSCs were differentiated into neural progenitor cells that gave rise to 2 distinct layers of the cerebral cortex. These cells were then suspended in a solution to create 2 separate bioinks, which were subsequently printed to produce a 2-tiered structure. Over a period of several weeks, the printed tissues demonstrated the preservation of their layered cellular structure, as evidenced by the presence of biomarkers that are specific to each layer. After the implantation of printed tissues into mouse brain slices, there was noticeable and cohesive integration, evident through the neural processes projecting and the neuronal migration across the boundary of the host implant. The implanted cells also exhibited active signaling that corresponded with the signals of the host cells. This communication between the human and mouse cells demonstrates both functional and structural integration. The scientists have plans to fine-tune the droplet printing process in order to fabricate intricate, multi-layered cerebral cortex tissues that more authentically imitate the structure of the human brain. Not only do these engineered tissues hold promise in the realm of brain injury recovery, but they also have the potential to be employed in drug testing, research on brain growth and development, and to enhance our comprehension of the underlying mechanisms of cognitive processes. The most recent breakthrough builds upon the team's extensive 10-year history of inventing and securing patents on 3D printing techniques utilized for the production of synthetic tissues and grown cells. According to Dr. Lina Zhou, the senior author from the Department of Chemistry at the University of Oxford, their methodology for printing droplets offers a method to create 3D living tissues with specific structures. This development brings us a step closer to creating individualized implantation treatments for brain injuries. Associate Professor Francis Seale, the senior author from the Department of Physiology, Anatomy and Genetics at the University of Oxford, stated that the utilization of living brain slices is an effective technique for exploring the potential of 3D printing in brain restoration. By studying the development of 3D printed cortical columns in vitro and their subsequent integration into brains during animal models of injury, this method serves as a natural link between these two processes. According to Professor Zoltan Molnar, who is the senior author and a member of the Department of Physiology, Anatomy and Genetics at the University of Oxford, the process of human brain development is intricate and fragile, with a complicated choreography. It would be foolish to assume that we could replicate the entire cellular progression in a laboratory setting. However, our 3D printing initiative is a significant step forward in managing the destiny and configuration of human-induced pluripotent stem cells, iPSCs, to establish the fundamental functional components of the cerebral cortex. The ambitious project was made possible thanks to the collaborative efforts of multiple fields, as fostered by Oxford's Martin School. The project's lead, Professor Hagan Bailey, who teaches at Oxford's Department of Chemistry, expressed that this futuristic undertaking was only achievable through the combined contributions of the Department of Physiology, Anatomy and Genetics at Oxford. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.